Hello and welcome to this first video on chapter 42 where we're going to start talking about the immune system. And in this first video we are going to discuss section 42.1 on innate immune response. Our learning objectives are to describe the physical and chemical immune barriers, explain immediate and induced innate immune responses, discuss natural killer cells, describe major histocompatibility complex 1 molecules, and summarize how the proteins in a complement system function to destroy extracellular pathogens. Before we dive into detail, I wanted you to get a good overview of our immune system. We break the immune system down into innate immunity and adaptive immunity. This section is focusing on innate immunity, where we have a ton of components that are going to protect our body through an array of substances. And we've got our skin and mucosal membranes that prevent entry, and some non-specific internal defenses, such as having immune cells, like our macrophages and NK cells, chemicals, such as interferons and complement proteins, and physiological responses, such as inflammation and fever, that can ward off these pathogens. Adaptive immunity will be discussed in section 2. And this is where our lymphocytes, our B and T cells, are going to be activated and replicated to respond to a specific antigen. It is tailor-made and therefore it takes a little bit longer for this immune system response to come about. We divide it into two further subdivisions of our immune system. Our cell-mediated immunity involves our T cells, and our antibody-mediated immunity involves our B cells, which once they come into contact with an antigen and become activated, they will differentiate into plasma cells, which are going to make and secrete antibodies. So keep this figure kind of in your back pocket to reference to when you feel a little bit lost. It'll kind of help to bring you back to having an understanding where we are in the immune response. So now let's get into our innate immunity. It's going to occur naturally because of the genetic factors and our physiology that is put in place when we are born. And any pathogenic threat is going to trigger this consistent sequence of events that can identify the type of pathogen, and this can be induced by infection or a vaccination, but it works to reduce the workload of our adaptive immune response. So first, let's get started with our physical and chemical barriers. Our physical barriers, of course, will include our skin. It functions as a continuous, impassable barrier to potentially infectious pathogens, and they can also come into contact with our skin, the pathogens can, and be killed or inactivated. This can be done through desiccation, which means drying it out. We know that the top layer of our skin is dead, so when they flake off, the pathogen can kind of leave with it. Acidity, due to the secretions of our skin oils from the sebaceous gland known as sebum. It creates an acidic mantle that some pathogens cannot withstand. And we have beneficial microorganisms that can cause something like microbial antagonism, meaning that the microorganisms on our skin can coexist with us because they are helping us outcompete the pathogens. They take up real estate and prevent infection from taking place. We also have our mucosal membranes that line areas like our digestive tract, respiratory tract, and our urogenital tract, and areas of our eyes. Our eyes can use tears, whereas mucous membranes can utilize the mucus secretions to trap and rinse away the pathogens. We also have cilia in certain areas like our nasal passages, pretty much through a majority of our respiratory tract, that can help to push the mucus with pathogens out of our body. We also have areas like our stomach that have low pH, and as we said before, when we create an acidic environment, certain pathogens cannot withstand that and will end up dying. Then we have our blood proteins that can bind and disrupt bacterial cell membranes, and urination to help us flush those pathogens away from the urinary tract. Now here you could see some of our innate offenses, and now we're going to start talking about some of the chemical barriers that can take place. These barriers to entry were mainly our physical barriers. We also refer to this as our first line of defense. Now with our chemical barriers, our pathogens have entered the body, but we are going to use certain processes and cells to keep them away. So we can produce inflammation. That's kind of the process that we are seeing over here with our inflammatory response. And we can use some of our phagocytes, like our dendritic cells, to engulf the pathogen. And with that, we're going to talk about some more details in how they will work with our adaptive immunity to trigger an even deeper immune response. And we also have secretion of some protective proteins, such as our complement proteins and interferons in plasma. All right, now let's talk about how we're going to recognize a pathogen. We have these pathogen-associated molecular patterns, and they can really be a variety of things, especially depending on what pathogen we have. Is it a virus, bacterial cell, a protozoan, and so on? But we can essentially recognize maybe carbohydrates, polypeptides, and nucleic acid signatures that are expressed by these pathogens. You can see some examples over here where we have certain structures on the pathogens, like a lipopeptide or a flagella or a peptidoglycan cell wall and so forth, that could react with what we call pattern recognition receptors, or PRRs. And these are the molecules that we find on our macrophage and dendritic cells that are going to be able to trigger an immune response. Now notice how some of these receptors are named TLR2, TLR5. That stands for our toll-like receptors, and they're a type of PRR that recognizes molecules that are shared by pathogens but distinguishable from the host molecules. Our next slide here is a summary slide of all of the innate immune cells. We are going to be talking about these one by one, so I won't bore you by reading off each of these slides. You'll kind of see their functioning and where we find them throughout the rest of this lecture, but this is, again, a good table or figure to come back to in order to clarify exactly where this is happening and what their functioning is. Now, a lot of our immune cells are going to be able to release something called cytokines, especially when we have a binding of those PRRs with the PAMPs. And a cytokine is going to be a chemical messenger that will regulate cell differentiation, basically dividing and becoming a specific type of cell, proliferation, which is more like cloning the cell, and gene expression. A subclass of a cytokine is an interleukin, and they're going to assist us in signaling that a pathogen is present and needs to be destroyed, along with any cells that have been infected with a pathogen. So it really helps to bridge the gap between our innate and adaptive immune responses. These were named so because they mediate the interactions between our white blood cells. Now you may be wondering, where are these cytokines released from? It's the infected cells that can release them, although we can also have immune cells release some cytokines, but they travel from the infected cells and go over to uninfected cells and induce those cells to release cytokines. And so, as you can imagine, we have lots of cytokines being released everywhere, and that is called a cytokine burst. Now, another subclass of cytokines are interferons. Interferons are released by the infected cells so they can go over to their neighboring cells that are uninfected and tell them to inhibit viral replication. Let's back up a little bit and talk about what happens when a virus infects a cell. A virus is first going to attach to that cell, It then enters the cell through endocytosis, and essentially that virus is going to hijack our cell's machinery and use it to replicate itself. So it creates lots of copies of its genome, it creates its viral proteins, and eventually it takes all those components, reassembles them to become multiple viruses, and eventually if we get enough viruses building up within a cell, the cell can burst and release more of these viruses that can then go and infect other neighboring cells. So like we said, what an interferon does is when a cell is infected with a virus, it is going to travel to the neighboring cells and tell it to turn on antiviral genes so that it doesn't allow the virus to hijack its machinery. That then reduces the protein synthesis and triggers our infected cells to go through apoptosis. We can also activate our nearby immune cells. Next, let's walk through the process of phagocytosis and inflammation. This is now our second line of defense because the pathogen has been able to get through those physical barriers. So we're going to employ our neutrophils, which are the first white blood cells to arrive, and also our macrophages and their phagocytosis processes to gobble up any pathogens that have entered into our tissues. We have 4 hallmark signs of symptoms, which are redness, heat, swelling, and pain. Now there actually is a 5th one as well, known as loss of function. Now notice how we are going to have a mast cell in here that resides within the tissue. So as soon as it senses that there's some type of damage to the tissue, it's going to release histamine, and histamine is going to dilate our nearby capillaries, allowing us to increase blood flow and increases the permeability of them, meaning we're going to make them leaky so we can allow our white blood cells to move out of the capillary and into the tissues. So that's what allows our neutrophil and our macrophages to come to our rescue. So let's talk about these hallmark symptoms. We, like I said, are going to vasodilate our blood vessel, and that is going to increase our blood flow to this area. And because we have increased blood flow, we will see our skin redden and become warm, hence the heat. So it increases the temperature, which helps to inhibit the growth of the pathogens that are present. Also, the increase of this blood flow is allowing more of our white blood cells to move through the capillary and into our tissues. And that also causes our proteins to escape into the tissues, and we can get blood to clot there, which can prevent us from losing our blood. Now, because we have so much excess fluid within this area, we're going to have some swelling take place. And within our skin, we have free nerve endings. And with all the fluid kind of pressing upon those free nerve endings, that is what causes the pain. So we kind of talked about this already, but let's talk about our white blood cells moving through the blood. Our capillary has increased permeability, so that's allowing these neutrophils to squeeze through and make their way into the tissue so they can phagocytize the debris, any dead cells, and bacteria that are found in the area. And they can usually localize any infection and keep it from spreading to other areas. They do die off very quickly and become a white or yellowish substance called pus. Now, if those neutrophils become overwhelmed, they're going to secrete some cytokines, and the macrophages are going to come to our rescue. If you remember from our blood lectures, we've talked about how in the blood they are called monocytes, but once they move into the tissues, they are called macrophages, and they are very powerful phagocytes and can gobble up the largest substances.